Bismillahirrahmanirrahim. My name is Humaira Amin and currently I am working as lecturer at the Department of Genomics and Bioinformatics at Cholistan University of Veterinary and Animal Sciences, Bahawalpur. Today I am going to explain about clinical exome sequencing and its application. First of all, I will introduce about the exome. Exome is actually the protein coding region of the genome. As we all know that the complete genome consists of the protein coding regions as well as the non-coding regions. Protein coding regions are known as exomes, while the non-coding regions are known as introns. And these protein coding regions consist of only the 1 to 2% of the complete genome, and while the rest of the genome consists of the introns. And these protein coding regions are very crucial because they contain the instructions for the proteins. So the exome sequencing is actually the technique which deals with the sequencing of these protein coding regions. And the difference between the whole genome sequencing and exome sequencing is that the whole genome sequencing captures both the intronic as well as the exonic regions of the genome, while the exome sequencing sequence only the protein coding region of the genome. So the sequencing process consists of 4 major steps. In the first step, DNA extracted from the sample, which may be in the form of blood or tissue, and then in the next step, library is prepared in which the isolated DNA is sheared into random fragments. Then the adapters are attached to those sheared fragments of DNA, and then in the next step, the probe method is used to capture the exons, and then after the capturing the exons, the nucleotide sequencing of the exonic region is performed. Major advantages of clinical exome sequencing are the comprehensive coverage of clinically relevant region, because it mainly focuses on the mutations which are crucial for the health and diagnosis. And it is cost effective because only a small portion of the genome is being sequenced, and the non-coding region, which is comprised of the major portion of the genome, is not being sequenced. Speed and efficiency, because it gives the faster turnaround time for the identification and diagnosis of the disease. Applications of the clinical exome sequencing include the personalized medicine, because specific treatment may be given to the person based on the known genetic information of that specific patient. And then pharmacogenomic, it has also role in pharmacogenomic, because we can predict the drug response of a specific patient based on its genetic makeup. Cancer genomics, clinical exome sequencing also has application is cancer genomics as well, because we can use this technique to identify the somatic mutations. Somatic mutations are those mutations which are mainly in the somatic cells of the body, and then those somatic mutations will be captured, and then we can diagnose the certain types of cancers like breast cancer, colorectal cancer, lung cancer, etc. It is also application in prenatal and neonatal applications, because our fetus can be sequenced by taking its sample to identify any of the birth defects, and then the neonates can also undergo this type of sequencing to identify the treatable genetic disorders. And the examples include phenylketonuria and congenital hypothyroidism. Carrier screening can also be performed using clinical exome sequencing, because it can identify the patients or the persons which contains the mutation for some specific recessive disorders, and it will help them in their reproductive decisions, because some of the diseases, some of the mutations are linked directly to the homozygous or heterozygous recessive conditions. And if that person is aware of the recessive mutations which are present in his or her genome, then it will help them in their family planning. And these examples of such disorders include the cystic fibrosis and sickle cell anemia. Challenges and limitations include because it may miss the coverage of some of the exomes, and coverage, that's why the coverage gaps may remain, and then the diseases, some of the diseases may remain undiagnosed. The second one is that because this, clinical exome sequencing mainly captures the single nucleotide polymorphisms and angles, but it lacks the detection of the certain types of variations like structural variations, copy number variations, insertions, and inversions and translocations. So the diseases which are caused by such type of mutations may remain undetected. And because this does not include the sequencing of non-coding regions, so there are certain diseases which are diagnosed by capturing uh, the mutations in the intronic as well as the exonic regions. And if we know we will not have the information of the intronic region mutations, then we cannot diagnose that disease. That's why it is the major lacking. And then the variant interpretation, there are certain categories of the variants for which the clinical significance has not been assigned, and that's why the variant interpretation sometimes cause the major problem in the diagnosis of the disease. Future directions, future directions include that we can improve the sequencing technologies to overcome the coverage gaps issues, and then we may integrate this technique with other omic techniques like transcriptomics, proteomics, and metabolomics to find out the expression of those genes, and then at the protein level and then the metabolome level as well, to find out the functional effects of that specific mutation. And we may need to expand the awareness to the clinicians to focus on this type of sequencing to have help in the diagnosis of the certain diseases. So this was all from my side.